This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, a special episode recorded on June 9th, 2018. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Today we are recording in Atlanta, Georgia at ASM Microbe 2018. And my guest is a professor of biology at the University of Puget Sound in Tacoma, Washington. Mark Martin, welcome to the show.
It's a pleasure to be here, Vincent.
Known you for quite a few years now, and it's about time we talked. On record, anyway. We always talk, don't we?
That's correct.
Let's start out by finding a little bit about you. Where are you from originally?
I was born in Compton, California, which surprises people. I am straight out of Compton. But, well, it's true. I have the birth certificate to prove it.
We believe you.
And I was raised in North Long Beach. And so I am a native Angelino.
I'm just pausing to take a picture of this little setup here, which you have your microphone. There's an animal that you've placed on it. What would that be?
Well, that would be Gordy the tardigrade.
We'll get to tardigrades in a bit.
Okay.
I do have a tardigrade question for you. So you grew up in California. Where'd you go to college?
I got my bachelor's degree at UCLA, and my first microbiology professor was Sid Rittenberg, who you might know of.
Yeah.
And but I, at that time, took a general kind of cell and molecular biology degree.
And what made you want to be a bio major at all or science major at all? Do you know?
Sure. I know exactly why. And there are two basic reasons. This is something I'm talking about on Monday, but one of my brother's good friends, Terry Royal, rest in peace, had Duchenne muscular dystrophy, and he didn't have the use of his hands. And I was his hands. I don't know if I have the plurals are correct. Anyway, I acted as his hands, and he loved science, so I was his lab assistant for several years. And he was the one who introduced me to Luminescent Bacteria. I remember that very clearly. And then later on, I did a science experiment with the memory of flatworms, planarians. And I remember at the time there was this fellow who claimed if you trained a planarian to run a maze, a T maze, and you chopped it up and fed it to an untrained worm, it would then know, which the science fiction lover in me adores this idea. And I found out it's very hard to build a T maze for flatworms. So I did teach them a conditioned response. Normally they prefer a dark substrate. They live under rocks and fresh water. And I taught them to associate that with a mild electrical shock, to the flatworms. And then what I was able to show is that when you cut them in two, the head half with the brain would grow a tail. The tail half without a brain would grow a head, but both sides remembered. And I won a pretty important award at the time because of this. I first generation to college, my father was a firefighter and my mother was a homemaker, and nobody was interested in science but me. So I was kind of on my own. I wasn't subscribing to Scientific American. I didn't know about the holistic theory of memory. But what I do know is when I won this award, I would get letters from people at Caltech saying you should consider a career in science. And that's when I first thought a person born where I was born in the socioeconomic status I had could be a scientist.
And why did Caltech tell you that?
Because the fellow who worked there, whose name's escaping me at the moment, it'll come to me later, actually worked on the holistic theory of the brain.
Got it. All right, so we've got you graduating from college with a science degree. What did you do next?
Then I went to graduate school at Stanford, and I was Sharon Long's first graduate student. I worked on the association between Cynorhizobium melilodi and alfalfa Medicago sativa. I also got a chance to meet Alan Campbell and Dale Kaiser and Charlie Yanofsky and all the rest. It was a very interesting time to be there.
Kornberg?
Oh, I met Art.
Very interesting place, wasn't it?
Yes, it was, and I was completely out of place. I have to tell you, I was Mowgli the wolf boy in this environment. But as I often say, I kind of apologize for my 20s. But I eventually grew up. But it was such a wonderful opportunity.
So, at that point, did you have some idea what you wanted to do?
My last year of college, I really fell in love with microbiology. Gary Wilcox taught me about the Arabinose operon. Peggy Cotter swears up and down that she knew me when she worked in Gary's lab, and I have no memory of it. So I'm glad she thinks of me fondly. So there you go. And I knew that's kind of where I wanted to be. I learned about phages from Bob Romig. It was a lot of fun. And then I had an opportunity to kind of meld my longstanding interest in symbiosis with microbes. And Sharon's lab had just started off. She just led Fred Ozabel's lab. And my specialty there was pretty much working on bacteriophages that infected Rhizobium to develop a transductional system to move genes along. It's very antediluvian now.
But useful.
Yeah, very much so.
So what did you do next?
So after that, I did a short postdoctoral stint in San Diego working on luminescence with Mike Silverman. Mm-hmm. And this is, again, I didn't have any external funding, so that was a very short period, and I'm very grateful. And then from there, I did, again, by that time, I hate to be personal, but I was married, and my then wife really liked living in San Diego. I actually did some work with Nigel Crawford on Arabidopsis.
I know Nigel.
Do you know Nigel?
He and I were in the same lab together. He was a student. I was a postdoc.
Interesting.
So he was a student with David Baltimore.
Yes, that's correct.
And we had the same bay. We used to talk a lot. And I left, and I kind of lost track of him. I know he did a postdoc in Arabidopsis, and I heard that he eventually got a job at UCSD, right?
That's correct.
I saw him about a month ago at David Baltimore's 80th birthday celebration, which was at Caltech. And get this. Nigel is younger than I am. He's retiring. I said, why do you want to retire? He said, well, I want to do some other things with my life. So
it's great work if you can get it.
I can't argue with that, right? Why not experience varied things? But that's funny. Yeah, Nigel and I used to talk on a daily basis. We don't always agree. He was philosophically a bit different from me, but he's a nice guy.
He was. He was very patient with me. Like I said, I've been very fortunate with that.
So you learned Arabidopsis from him, right?
I did.
Okay, and that was this short stint of a postdoc.
These are maybe a year and a half long each.
Okay.
And this is, happens to a lot of people in my position. And then I had an opportunity to join a biotech company in San Diego.
What's the name of that one?
And that was called Kelco. It was later purchased by Merck & Company. And I worked on bacterial polymers. I've always liked undomesticated microbes. That's my thing.
What does that mean, undomesticated?
They're not lab rats.
Like E. coli, K12, right?
Yeah, and I'm not upset about K12. I'm not upset about Salmonella or B. subtilis. I think they're wonderful. But I like the weird ones. I always have. And so as a result, I was working with some of the polymers like Xanthomonas campestris makes xanthan gum that's in a lot of like salad dressings. Also used in oil fields of all things. And then also another polymer, it's Pseudoultramonas elodie, made something called Gelan gum, which is in snack foods and is an agar replacement, which sounds like the beginning of a joke, but it's quite true.
Sounds good to me.
Yeah.
So what's next? What happened? You were at this company for a few years?
8 years.
A long time.
A long time. And so then there was some downsizing and I got laid off. And I don't know if you care about this, but I had published a couple of science fiction novels. And so I had.
Really?
Uh-huh.
I didn't know that.
Yes.
I know you're a Lovecraft aficionado, as am I. I didn't know you wrote science fiction.
I did. A very good friend of mine, Gregory Benford, is a physicist and a well-known science fiction writer. And he always felt that I should write. And so he took me under his wing, so I was able to get decent contracts. And so I had a little money from that, but I didn't really know what to do.
But these books were published and sold, so if we look up them, we could find them maybe?
You could. They're out of print, but, you
could
probably
find
will
them.
read some unpleasant reviews. To my mind, was it Robert Heinlein said the most elegant language that you can find in English is pay to the order of, followed by a number, right? I enjoyed writing them, and it served an important purpose in my life. I'm very grateful.
So you wrote those while you were at the company, right? Mm-hmm. Cool. Didn't know that.
And after I got laid off, what was I going to do with myself?
Mm-hmm.
And a friend of mine from graduate school, Daniel Kleonsky, I don't know if he's—
I don't know him, no.
He's at, I got to get this right, University of Michigan. I can't say Michigan State. And he does work on autophagy over there. And
Or autophagy.
Oh, he's very specific.
He says autophagy?
It's a big deal to him, and I just don't want to fight with anybody.
What's the other word that's programmed cell death? What would he say to that? Apoptosis or apoptosis? I always have these discussions. Okay.
Well, he sent me an ad for a teaching job, a short-term teaching job at a place called Occidental College in Los Angeles.
I know it. I've heard of it, yeah.
I got a short-term position there, and then I got a tenure-track position there, and I worked there for about 8 years.
Teaching?
I did, teaching a small amount of research, and that's where I started working on Della Vibrio and assorted bacterial predators. And then that job didn't work out, as sometimes happens, and I was fortunate enough to get a job at the University of Puget Sound, where they did, in fact, award me tenure, and there I am.
How long have you been there now?
I think 11 years, 12 maybe.
So does it bother you when people say the state of Washington or Washington State instead of just Washington?
It doesn't bother me.
Some people it bothers greatly.
Yeah.
You know Mark Crislip? Have you ever heard that name? He's a podcaster. He has a podcast called the Infectious Disease Puscast.
Oh, my.
And he always rails against Washington State. He says it's Washington and Washington, D.C. Anyway, Puget Sound. Now, you said 18 years you've been there?
Oh, 12. I'm
12
sorry.
years. That was my mistake. And there you've been teaching and doing research, right?
I have.
Which I would like to talk about. So I went to your website, and I note the courses that you were teaching. Quite a few. 1, 2, 3, 4, 5, 6. Is that right?
Not all at once.
Of course not. Different semesters, different years.
That's correct.
Right? So Biology 111, Unity of Life.
Yes.
Did you make that title up?
No.
It was already there?
It was there when I arrived, but when I was at Occidental College, I also taught an introductory course, much the same type. So I actually like that. I teach it in the spring semester where I don't have as many biology majors because other majors require some kind of science requirement, and word got around that they should take it with me.
Yeah.
And so, but I have to teach it the same way. So it's pretty much an introduction to cell and molecular biology.
So who are the students mainly? Are they students that want to go to medical school or not sure or want to do science? Do you have some sense of the makeup?
You mean in my department as a whole?
No,
or
in the courses that you teach. Yeah.
So in Biology 111, I would say half of my students want to be biology majors, small percentage are chem majors, psychology majors, and a few exercise science.
And no medical school?
No.
That's interesting.
Undergraduate institution, 3,000 students total at the institution, maybe 3,500.
No, I didn't mean that it was a medical school, but do any of them want to go on to medical school?
I'm so sorry for misunderstanding you. Sure, I would say probably about 20%.
Yeah, so I figure. At Columbia, it's like 50% in my virology class, but a lot of people want to be doctors. We know why, right? But there has to be more than that. And there's some people who want to do research. I guess you get you have people who go on to get PhDs, for example, from these biology courses.
That's kind of my specialty is directing that.
Yeah.
That's a big deal to me. And so, in fact, when I was walking around the floor just a little while ago, I ran into 3 students that I'd never had in class, but who knew me, who were all here working on their PhDs in microbiology.
So Unity of Life is kind of like an introductory survey of biology. You do microbes, but you do planaria and other things as well. Is that right?
Well, mostly, I mean, the way that we look at it, we have a series of labs that are being taught in multiple sections of this course.
Yeah.
And I surreptitiously will put in some microbiology, but there's a limit to how much of I can put in there. So I'll put in a little gene regulation. I'll put in a biofilm lab that I'm very happy with, that kind of thing. And we do a lot of PCR. We actually did the PCR experiment with PTC testing this semester, which was a lot of fun.
Cool. Then you have microbiology.
Oh, I do.
Is that your favorite?
It is.
And then you have another one called Symbioses and Parasitism. That must be a close second, right?
Well, I enjoy that, and I teach it in two flavors. When I was awarded tenure, I was told I had to now do more for the institution, which I thought was fun. And they said, we want you to teach a freshman writing class. And I said, I can't teach writing because it's very formulaic, as I'm sure you know, in the university or college system. And they made the mistake of saying whatever I wanted to do. And then I said, well, I want to make it about symbioses and parasitism. And they said, oh, that class won't fill up. And I put my hand out and I said, I'll bet you $1,000 it will fill up the same day you post it. And I should point out that my youngest son says that dad bets, but he never gambles.
That's great. I love that.
And it fills up every year. And they're first semester students. And they're wonderful young men and women. They're wonderful, wonderful people, but that's a big adjustment.
Sure.
So we will have, for example, in that class, I've had Ed Yong Skype in once. That was wonderful.
Yeah, right.
Irene Newton is Skyped in to talk about Riftia. It's a lot of fun.
I'm sure you make it fun. Is there a course you would like to teach but don't at the moment?
I would like to teach the symbiosis course with a lab.
I see.
I think that would be good.
Okay.
When I was at Occidental College, I would teach the introductory course in one semester every year, and then the other semester I alternated between genetics and microbiology. So I'm delighted to be teaching microbiology every year, but I have to teach an additional course now. And so it would be wonderful if I could kind of trade out from the introductory course and teach another specialty course. I would love to teach a virology course. I know how much you would appreciate that.
I would, and you could use my lectures as a guide to sort of help you construct the course, right?
Yes.
And that's why they're there. Many professors at undergraduate institutions tell me they use them to help not only design their course, but to learn the subject as well, right? Because unless you do virology, you just don't know all of this, right? So you should do that. And if you do, I can help you. I can give you all the slides and anything you need.
That's very kind of you.
So how do you keep the students interested in all these science courses? I know you do a lot of creative things because I see you online. I see you on social media. I see you on your blog explaining what you do and wonderful artworks that your students make. So how do you get them motivated and interested in the subject?
One word, it's ownership. A lot of students are passive. I think it's part of our media culture. They're staring at their phones just like I do. They're staring at a screen just like you are. That's how everyone is. And take a breath and ask them, what are you good at? What do you enjoy?
Yeah.
And what's interesting is that if you give them an opportunity to express themselves not in a way they're used to, say filling in a bubble on a form or answering a short answer on a test, they do amazing things. And can I give you a couple of examples?
I would like some, yes.
So in my introductory class, I had a young woman who was just deathly afraid of intermediary metabolism. And I'm not teaching it at a very, high level. I'm teaching concepts. I describe it as you need to have like a holiday tree, and then you later put all the ornaments on the tree. But you need to have that tree first. And a lot of times when you teach a survey course, they kind of throw it all at you at the same time as a student, and it's hard to make it gel. So for me, having some kind of central structure is really important. And that's what I told the students, and that's the best way to learn things. And so when it came to central metabolism, this young woman put together a board game. And this board game is called The Game of Life. And it was amazing, Vincent. I mean, there were pieces and cards and rules. And I have a picture of it up. I'll send it to you if you'd like. It's in my lab on the wall. And that young woman did really well on the final exam because historically the metabolism exam is challenging. Well, she owned it on the final. And it's because she had a different way of looking at the questions and how to put it together.
Did you suggest she make a board game, or was that her design?
That's all her.
You didn't even say do something different. You just let her go, and she just did this, right?
So that's correct. But you have to be careful. You just can't turn it loose.
Yeah.
Do you have the experience of the evils of the first draft?
Of course, yes.
And you and I say first draft, and what students hear is whatever I put on the page. And that's not what a first draft really is.
Right.
So the only answer to that is structure. And so I scaffold things pretty carefully. So they have to meet with me to talk about their creative project, and we talk about what's possible and what's not. I might make some suggestions, and they might spark off some ideas from what I'm saying. And then what happens is that they have to give me like an outline or a couple of paragraphs a week later. And then that gives them a direction to go when they finish it. And that seems to work pretty well. Some students choose not to do it. I can't really – I could require it, but I don't feel quite right doing that.
Another example.
Certainly. Another example I was trying to come up with. I had a student who actually did not go through the process and said to me, and I can show you a picture of this as well, that she'd made something for me that helped her during ceramics class. And I said, what do you mean? And she goes, I'll bring it. And she takes off her backpack and she brings a huge sculpture showing bacteria with the 3 different shapes that you find in bacteria, a phage on there. And I said, how did this help you? She goes, well, I would look at it and it would remind me of what was inside each thing. So it's a different way.
Right.
I think a lot of the students, again, are too passive, and it could be actually building the things is how that works.
I like that. And a lot of these are on your blog, right?
Yeah.
microbesrule.blogspot.com if people want to check it out.
I need to, of course, update it. This is something you've been telling me for some time.
I did.
Yes, and this is why. And that's something I'm going to be doing this summer because I do think it's important to get that out there and the different things that people have done.
Well, it's a good example of what you're doing. You have a lot of photographs of things your students make.
Yes.
There's one thing that someone made. Let me look at it here. It was and of course they thank you. It's lovely. And the buttons. We'll get back to. Here we go. Doc Martin, thank you. The microbial supremacy tree of life. Right.
Oh, this is Mariko's work, right?
Yes.
Mariko Hunter.
Mariko, right.
This is a really important story to tell. And if you look here, you'll see there's a particular button that I'm wearing.
And she's holding it here in the picture.
She is indeed, because she came up with it. Mariko was – I don't think that she was terribly – she was afraid of the class. She thought it would be too difficult. There's often a dichotomy at undergraduate institutions between people who do work in – I hate the expression big and little biology. I really hate that. But certainly cell and molecular versus evolution and population biology. And I think they're all part of biology. But that dichotomy exists regardless of what I wish. So Mariko wasn't sure she could get through this course. And I noticed that she did cartoons on her exams. And they were always relevant and they were really well done. And I said, are you an artist? And she goes, oh, I just doodle. Now,
that's what Picasso said, right?
Right, yeah, that's true. But it's interesting to me because at first I thought, well, maybe she's just being very modest about it. But I honestly think many of these students don't appreciate their skills.
Oh, yes, totally.
And so on the last day of class, after she turned in – well, not the last day, the day of the final. After she turned in her final, she handed me a folder, and that's what you're looking at there. And that folder is this drawing of all the things that I talked about in class. There's an attenuator, if you see for the trip operon there.
Right.
There's a Petri dish with shoes because I've been known to call people Petri dishes with shoes. There's the tree of life itself. There are many other things there, but over in one corner you'll see a rocket ship.
Right.
And it says, Micronauts we soar.
Exactly.
And I would call them all Micronauts.
All the students.
Yeah, because you're now my Micronauts. And
lovely.
And in fact, this has become like buttons that I make and emblems. And I think it's a good way to look at it. And I will tell you that when you do the stuff like with the buttons and the T-shirts that I've done, it creates a sense of camaraderie in the classroom.
So this particular one, Micronauts we soar, you had Vexed Muddler make it in a cartoon, and then you had a button made.
I did.
And it's beautiful.
Yeah.
It's beautiful. It says Micronauts we soar. We have a rocket, and in the background are microbes. And so you gave it to the students in the class?
Yes.
It was made in time for that, right? Now, you also make other pins.
I do.
As you've just said. I have some here. You used to have one called Microbial Supremacy, but you said people don't like that word anymore, right?
Yeah, some people I really like and admire that it really disturbed them. And no, I'm not. I don't belong in this century, I often feel. And if it really bothers people, that's valid. So what could I say instead? May I explain where microbial supremacy came from?
Yes, of course.
So this is a story many people have to listen to from me, and I apologize for repeating it. But years and years ago at my current department, before a department meeting started, two of my colleagues who happened to be botanists were saying, if we get more money, what kind of tenure track position could we get? What kind of area would we want our scholar educator to be in, research-wise? And they're both botanists, so of course they felt that botany should be the area because it is, after all, the center of life, as they said. I didn't cough. I didn't roll my eyes. I just sipped at my coffee. And Betsy Kirkpatrick, in fact, who's since retired, said, well, Mark here thinks that bacteria are more important than anything else. Mind you, I didn't get involved in this. And I looked at her and I said, Betsy, I think plants are very interesting. It's just that all the really cool parts they have, they co-opted from bacteria and archaea. And she snorted with laughter and said, you, sir, are a microbial supremacist.
Wonderful.
And I said, yes, I am.
But they were here first.
Yes, and last.
And everything depends on them.
I agree.
So I would agree with you.
First evolved, last extinct, right?
I think it's great. I love it. I'm sorry that you're not making more microbial supremacy buttons, but you have some others. So you make these every year, and they're different, and you give them to the students?
Yeah.
At the beginning, middle, and when?
So I have the buttons the first day of class. I also I make themed candy for them that has sayings on it. It's
T-shirts also?
T-shirts are later in the semester. I want to see if they're going to drop first.
Yes.
Because guess who's paying for this? That would be me. Right?
Yeah, of course.
And Cafe Press, and I'm not taking a position on it. There are other people, other vendors that do this stuff. It's just what I've used. And I do it every year. I usually work with an artist and a science artist. And I've worked with Caitlin Reese, who's done a lot of my earlier stuff. My wife, Jennifer Quinn, has done some. And the artist currently known as Vexed Muddler has done a lot of my stuff as well.
Right. Vexed, Muddler, and Michelle Banks designed the logo for my little LLC, Microbe TV. It's very nice. And every year I see you, I get some of these buttons. And so here's some in front of me. We have Occupy the Curriculum. We are the 99.9999%. Those are the microbes saying that.
That's what they're saying. And they're saying no ukes.
Oh, I can't even see it. No ukes, right. And what's the other one say? Microbial supremacy now?
See, it still sneaks in, doesn't it?
It does. So what do you mean by Occupy the Curriculum? Is it like Occupy Wall Street?
Well, one of the things that I found, and I'll talk about this later in the week, but if you were to go to 100 people and ask them, what is your response to the word bacteria or the word germ? They have to give a one-word response. Some students are very resistant, or they'll copy the next person. It's hysterical to me. It's almost negative, almost always negative.
Yeah.
There's a very negative microbial PR thing going on. This is why I have deep concerns about what I call swab stories. We can talk about that later if you want to. So part of my job is to be I can't say the word prokaryote anymore. I used to call myself the prophet of prokaryotes, but I can't really say that. But the point is, I think that it's at the center of everything. And it's not original with me. There are so many famous people that have said the same thing. And I don't think students really appreciate that. When I tell them, for example, that rubisco and nitrogenase are bacterial enzymes, because they are, and we wouldn't have efficient carbon fixation, and we wouldn't have bioavailable nitrogen without them. I think Jack Gilbert and trying to think of the other gentleman, Josh Neufeld, did a wonderful article in PLOS about a world without microbes. And it really deals with this and other issues. And I find that very few of my students know this. In fact, Vincent, I don't know if you're aware of this, but many high school students aren't taught that mitochondria and chloroplasts were originally bacteria. It's just thought not to be terribly important when I think it is the important issue in cell biology. And so I am going to be fighting the fight. Now, I know that several people like Diane Newman, is she at Caltech or has she gone back?
I believe she was originally, yeah.
Okay. Well, Diane Newman, wherever she is, being fabulous. And then Margaret McFall-Nye, the University of Hawaii now. I know that they've done a lot of work with the Howard Hughes Medical Institute about trying to revamp the undergraduate curriculum to make it more microbially centric.
Right.
And I would be 100% on board with that, but that's way above my pay grade.
Diane Newman is at Caltech.
Good.
The power of Google.
Yes.
So these buttons you can also buy, right? People can buy them if they want.
If they wish to.
Where would they go to buy them?
cafepress.com/microbes rule.
Microbes rule. So I have here Micronauts we saw, so that's great. I have my mind on my microbes and my microbes on my mind.
Now, you probably don't know where that's from unless you're really part of the hip-hop community, Vincent.
I'm not part of that community.
So Snoop Dogg has a song called Gin and Juice.
Uh-huh.
And part of the song he says, with my mind on my money and my money on my mind. And based on that rap, I thought it would be funny to have with my mind on my microbes and my microbes on my mind. And then Caitlin Reese did what is meant to look like a brain.
Yeah, the squiggly red thing, yeah.
Right,
definitely.
with microbes. Yeah.
You think there are microbes in our brains?
You know, that brings up the whole kitome issue, whether or not we have things in the kit. But I find it very hard to believe there aren't some in there.
Yeah. I think they're everywhere, right?
Yeah.
Then another one, if microbial centricity is wrong, I don't want to be right.
I don't want to be right.
What's that from?
Well, that's the same one as supremacy, but with the more PC kind of title.
Yeah, I love these. I think these are great. It's a great idea. I should really do this for my class, but you'd have to plan ahead of time, I guess. And these are all working with your artists, right? Mm-hmm. Which is nice that you can do that. I guess I could do the same thing. Right?
And it's interesting to me, if you look at T-shirts and you just do searches on Google, you'll find that people steal images all the time. Mm-hmm. They'll steal my images. And, I'm not going to fight about things, but I think it's really important if you're going to use artwork to commission the artwork, which I always do, and to give credit for that artwork.
Sure. Do the students wear these buttons after you give them to them? Yeah, they do. That's great. Now, you're here because you're getting what's called a Karski Award.
Yes.
What is that for?
You must understand that my mother, rest her soul, was psychologically Amish, so that you can't really brag about anything. But what the award says is something along the lines of excellence in teaching microbiology and mentoring students in their future endeavors. And I am ultra uncomfortable saying that, but there you go.
It's all true, clearly. From what we've briefly discussed now, and people can go online and see for themselves, you're clearly highly deserving, so congratulations.
Thank you very much.
I think it's great. And the part that's hard, I guess, is that you have to keep doing this. Keep it up and get better and do more, right?
It's very interesting. I have always am thinking about what to do differently. Mm-hmm. I mean, in my microbiology class, for example, I'm thinking about trying different modules than I used to have a series of labs that everyone would go through, and then for the last 4 weeks they would have independent projects that they came up with. Mm-hmm. And I'm just not as happy with that as I could be. And even though sometimes the people that help me with setup aren't happy with this, as they would like to just do the same thing every year, I'm thinking about making some changes to it. You know, Vincent, there have been some wonderful things that my students have done for their independent projects. Mm-hmm. Do you know about pink pigmented methylotrophs?
I do not. Tell me.
These are bacteria that use methanol,
right,
which is part of plant biochemistry, plant metabolism. And these organisms are pink, hence the name, and they will live on methanol as a carbon source. And you can print a leaf onto this plate, and you will get the outline of that leaf in these pink organisms.
Nice. So you have the students do that?
I had one student do that. What is each student interested in? I remember these are not large classes. I have no more than 20 students in microbiology. Mm. And so that way I'm able to meet with each of them and talk to them more and find out what they're interested in.
I think that's great.
Yeah.
Right. So I do – here's what I do. Tell me what you think. I teach a virology class to undergraduates at Columbia, and I have about 90 students every year. So I have them do an extra credit at the end. They watch the movie Contagion, and I say, tell me 3 things that are wrong with it. And they really like that. And many of them say, I watched it 2 years ago, but it's totally different now that I took this course. And that's really the goal, right?
It is.
To get them to see it in a different light. So is that good? You like that?
That's wonderful. It's we used to do that in genetics. We would watch the movie Gattaca, which is a fun movie.
Yep.
What's right, what's wrong. Get them thinking critically about things.
Yep.
And this is very similar to what you were just saying. One of the fun exercises I give students is I tell them that most people, when they look at microbes in general, they originally were like devil microbes.
Sure, of course.
And I had Vext make me an illustration of what that would be. And, as all microbes are bad. And then you have other people that seem to go the other direction, angel microbes, when most microbes are like, meh, right? They're indifferent.
Yeah.
And so I'll have students go out into popular literature and find examples of each of these things. And as you say, they really enjoy doing that because their opinion matters. And they say, hey, that's not the way I learned it in class. And that's very important.
Is this a good time to talk about the swab story?
Whatever you'd like.
Because I want to next move into a little bit of research. Does this fit in at this point?
Yeah, you want me to talk about swab stories? I'm happy to.
Yeah.
So a swab story is basically, and again, I don't want to pick on people who publish in this area. If you look around, there are tons and tons and tons of articles about there are bacteria on fill in the blank. Your shoes, bottom of your handbag, toilet seats, toothbrushes, wherever it is. And it's buying into the idea that microbes are bad, when in fact that's not true. And what these people are doing is taking a swab, they're swabbing an area, and they're saying, oh my goodness, there are bacteria on that swab. It's so bad I want someone to do a paper on the bacteria to be found on swabs. And the whole problem is, how do you get around this? Yeah. And you find that people get really scared, and then they'll do things like, I've got to wash my hands, I've got to use antibacterial soaps and all the rest. So I send students out for looking for swab stories. You probably saw the one recently. I got quoted on this one where they were making hay out of the Dyson hand dryers, spraying things all over the room. Well,
right. I saw that, yeah.
What do you think happens when you flush a toilet?
Oh, yeah, big time aerosol.
Right. And you were talking to me about viruses being spread by the process of urination. I remember that from a couple of years ago. This is the kind of life we have, folks, talking about this. In any event, they enjoy looking for swab stories. It changes the way they view everything. And like I used to say, I wish I had microbe-colored glasses to have them put on.
Yep.
For that reason.
Yep.
So that's what swab stories are.
So you know what bothers me, Mark, is that often they don't even culture, they just sequence.
Yeah.
So what does that mean if you find pieces of bacterial DNA? Who knows, right?
It could be the necrome.
The necrome. I like that, the necrome. So there was a study done in New York City recently where they swabbed all over the place and they found all these bacteria, but they just looked for DNA. So who knows if there were actually replicating bacteria there, right?
I agree. And also the proportions. I don't know what the statistics are, Vincent, but if I did a swab of 100 people's skin, I'll bet you that I could find toxin genes from MRSA on some proportion of them.
Of course.
But the question is how many of them there are. And the question isn't answered that way.
You can't. You absolutely can't. So this brings up a bigger issue, is that issues with microbial reporting in the general press. I'm sorry you think about this. You may even have your students go looking for egregious examples, right?
That's exactly what we do with angel and devil microbes, where they actually look for these reports.
That's a course you teach? No. Angel and devil microbes. Oh, I see. That's the thing you told me before.
Oh, yeah.
Good ones and bad ones. Yeah.
So just having – and, to some degree I do that even with my first-year students when we are looking for the way things are being presented to them. Parasites, are they always bad? Well, you know better than that.
Right.
Talking about the hygiene hypothesis, that mean they're always good. Well, that's not true either.
Sure. Well, I don't think there's much we can do about it. What I say to my class is, some of you are going to be writers, fix it.
Yes. Vincent, one of the things that's funny to me is Caitlin Reese made me something really amusing one year. And it's a idealized set of hands holding hands with different spots of color representing microbes on them, right? And the logo, and I'm responsible for this, yes, is when we're holding hands, so are they, right? Great. Students invariably make gagging sounds and carry on about this. And then I flash the title to that microbiome paper about the number of microbes transferred during, and I love this euphemism, deep kissing. Mm-hmm. And I point out, no one's given up kissing.
Nope.
So you can't tell me you're grossed out by holding hands. So it's all about what you perceive as being dangerous, right?
Sure. What you can't see, you don't worry about, right?
And I also have my students in my micro class, we swab their reusable water bottles.
Yeah.
And there are lovely things that we found there.
Of course.
Beautiful colonies. And I'm not saying that they're friends, but they're just part of their lives. This is a microbial planet. We are swimming in a sea of microbes at all times.
I got an email from a listener, I believe in California, who had her students, maybe this was 5th grade, swab different parts of the school, and they streaked them out to see which was the most contaminated. And she sent me pictures of these lovely plates, potato dextrose plates. Mm-hmm. And they found that the gym mats were the most contaminated. And she said, we're waiting for the principal to do something. I love it, though. I love that you get engaged. But it's all about telling people it's not bad. Don't worry about it. But the press, of course, will make a big deal.
Absolutely.
Now, you do some research. On your website, you have 3 main areas. And Vibrio is one of them, 2 of them, 3 of them, actually. And Ensifer. What is Ensifer?
Well, so I can talk about my program in general because there have been some changes since that. I need to update
you have to
website.
Update it.
Yes. Okay. There's a surprise. So basically, I'm very interested in predatory bacteria, and the 2 types that I work at are Vibrio and also Ensifer. Vibrio, I think you already know about. Ensifer was discovered by Lester Casita in the 1980s. Mm-hmm. He was seeding samples of soil with Micrococcus luteus. He had a grant from the Department of Defense for this. I guess you can connect the dots. He was trying to determine how long the Micrococcus would survive in these different soil samples, and lo and behold, he found that there are many predators. And this is a particular type of a predator. It turns out it's very closely related to the fast-growing rhizobia, so it all comes back to, my old home week with rhizobium. And it's very specific, unlike Vibrio. It just seems to like Micrococcus luteus, and it does this fascinating thing where it tracks along a streak of where I've put Micrococcus. It doesn't do it to E. coli. It won't do it when the Micrococcus is dead. So we have a genome published on that with Dave Baltris and Laura Williams, and we're doing some transposon mutagenesis to try and figure out what's going on with that bug.
Mm-hmm. So you could mutagenize and see how they lose track, right?
Yes, lose track of tracking, yes.
And then see what genes are involved. That would be fun.
I'm very interested in that. Uh-huh. I also have a long-term interest in some of the symbioses that I find kind of in areas that my colleagues are involved in. And so, for example, in the Puget Sound area, there was a large wood industry, sawdust mills everywhere. Mm-hmm. So there are areas, Vincent, that are under the sand, 10 feet of sawdust. So the tide comes in, and then the tide comes out. So what this is a ginormous bioreactor, and it's fermenting the cellulose.
Sure.
And so this next part I want to be very clear on. The effluent, when the tide goes up, that comes out of that sodden area is 4 millimolar hydrogen sulfide.
Amazing.
That's 50 times higher than you find around hydrothermal vents.
Right.
And so around these areas, you find huge filamentous bacteria. And Joel Elliott in my department and I are trying to study these organisms, which seem very similar to what you find around hydrothermal vents. Mm-hmm. Students love this kind of work, especially because some of the crabs get covered by these same filaments. You are familiar, no doubt, with the Yeti crabs that are found around some of the hydrothermal vents. So I'm calling these Sasquatch crabs, right?
Wonderful. Love it.
But what's really exciting about my collaborations is with Stacy Weiss in my department. She studies a small lizard in southern Arizona, and we just got an NSF grant for this. Now, what we found out several years – well, back up. This is what happened. Stacy studies these lizards, and there's lots of reasons that she's interested in them, and I won't take up your time with those. But she said to me that when she dissected out the eggs versus having them naturally laid, the dissected out eggs tended to go bad. They clearly were infected by fungi or bacteria at a higher rate than the ones that were naturally laid. I said, perhaps microbes are being deposited on them that help protect them against pathogens. There's evidence for that among the birds, for example, and there are elements like that in humans even. But what we're dealing with reptiles is they have a cloaca through which everything goes. So we did cloacal swabs, and there's a lot of humor in that because these are not big lizards, right? I won't do the jokes about it, but the long and the short of it is they have very different microbiota. So the males, their cloacal microbiota seems quite similar to their gut microbiota, but the females tends to get them really streamlined at the time that they're starting to lay eggs. So what this NSF grant lets us do, and we're very grateful for it, is be able to do 12 months of the year to find out when these changes take place. My job is to look for antimicrobial activities from some of the isolates, and then use, functional metagenomics to see if I can find other types of genes that could actually be responsible for these antimicrobial effects.
Right. Now, how much do you devote to this research versus your teaching?
I would say it's supposed to be 75% and 25%.
75% teaching?
75% teaching, and then 25% research.
And is that how it is, or it varies?
No, it's usually 75% and 75%. That's the answer you're looking for.
Very good. And do your students participate in this?
Absolutely. We don't – it's one of the things that is expected of each of us to do active research with our students, and I consider it to be teaching.
Of course.
Because it is. And I work with each student, and I'm a little different than, say, my colleagues in the chem department because they'll have everybody attack the same kind of problem. I need to find out what interests the students. And, they might not know what interests them, and I'll get them onto that. But I've had a pretty good track record doing this. And so they learn a lot from that. Do I produce as much in terms of publications as I should? Certainly not compared to an R01 institution. But I have sent an awful lot of students to PhD programs because they learn how to think scientifically, learn how to ask questions and answer them.
So will you typically pick up freshmen and keep them for a couple of years? Is that how it works?
We more trap them, and we put we put like location beacons on them. No, I usually get them as sophomores if I do. Sometimes I get first year students, and that's great. In fact, one of my former students here, Kim Dill McFarland, was one of the ones that I had years and years ago. She's now Dr. Kim Dill McFarland. So nice to see them do this.
And they come from your courses, I presume.
Yes.
And they come up to you and say, "Can I do research with you?" And that's how it works, right?
I will. If there's a student, I like to put questions on exams that are kind of open-ended. I make them for extra credit because some students they have 3 other classes and all the rest, but some students really get engaged. And I can see the spark usually. Am I a great researcher? Probably not. Am I a great teacher? I don't know about that. What I am is a good talent scout, and I'm really good at that.
So you can see someone and you'll ask them, would you like to work in the lab? And mostly they do, right? Well,
I've had some that weren't interested, and that's okay. Right.
How many would you have in the lab at any given time?
4.
For undergraduates. So there are no PhD students. You don't have a PhD program in your
No master's program, no postdocs.
So you are an entirely undergraduate-driven research operation.
That's me.
And you can publish your results, right? And the undergrads are probably really happy to do that.
Oh, yeah.
And I bet you help them to write the papers, right?
Yes.
And that's a learning experience in itself.
Yes.
And for an undergraduate to do that, that's pretty thrilling, right?
And there are more and more venues for doing that. I mean, if you sit there and say, well, everything has to be published in JBAC-T, you're welcome, Pet Slosh. What I'm trying to say here is that might be hard to do, but there are ways to bite off a question that a student is interested in. There are a number of new ASM journals that will be helpful for that in that regard.
Absolutely.
And I'm looking forward to that, especially as the technology gets better and better.
And there's so many good technologies around now that you can use to answer some of the questions you're looking at, and they're not dangerous.
No.
what you're working on is safe for students to do. It's a great experience.
And everybody loves bacteria that eat bacteria. I mean,
that's
I do
great.
too.
The predators are great. I just think that's a great story, and understanding how they do that is great. But the 4 millimolar hydrogen sulfide, that's pretty cool too. The lizards.
I agree.
They have a lot of projects.
Yeah. So if you throw enough stuff up to the wall, some of it'll stick.
In many aspects of life, we do that. We do that in science communication often. We do that when I write grants. I do that all the time. I write lots and lots of them and throw them at the wall, and now and then one sticks. And that's the way it works. I think you have to. We're scientists. We should experiment, right? Who are your professional heroes?
Oh, microbial heroes. Do you know what shrinky dinks are, Vincent?
Shrinky dinks. No, I don't.
You have children?
I do. They're grown up now.
So these are the plastic things that you cut out and decorate and you put in the oven and they shrink.
That's right, yeah.
Shrinky dinks.
Yes, okay.
Vincent, you're a shrinky dink.
I am.
I have a mobile that my wife made for me of my microbial heroes, and you are one of them.
Really?
Yes.
Well, I didn't ask you that for you to say that.
No, there's quite a few of them.
Who else is on that?
Well, Sid Rittenberg, right. My first microbiology professor, Pat Schloss, of course, Joe Handelsman, who's just been fabulous, Heidi Goodrich-Blair. I honestly sometimes get directed toward those we've lost recently. And Abigail Salyers was very important to me, and Ed Ledbetter, very important to me. And you have to know that Elio Schechter is high up there as well.
Of course. I would guess that, yeah. Oh, this brings up something I skipped, the Luminescent Bacteria. And we featured one of your wife does these, right? Mm-hmm. Jennifer Quinn, right?
That's right.
On an episode of TWiM. Tell us about those.
Well, as I mentioned before, from the time I was, I guess, 11, when Terry Royal sent me or brought home this plate that he said, go look at this in the dark. And I realized it wasn't some like 1960s glow-in-the-dark blacklight thing. It was actually glowing, and it had that wonderful kind of blue-green glow, which is because that's the wavelength that light travels best underwater. Right. So it makes perfect sense in terms of evolution. And I've always been playing with bioluminescent bacteria, but I wanted to have some kind of venue where students could play around with bacteria in a way that didn't sound dangerous.
Right.
So many people do demos where they'll say, well, what's living in hamburger? Or, let's see what's on your skin or whatever. I want to do something that it's something that's beautiful. And I mean, I did a postdoc in bioluminescence, so I kind of like the area. So this is a particular strain of luminous bacterium, Photobacterium legnothi, and it was isolated by Eric Stabb and Ned Ruby from Kaneohe Bay in Hawaii. Just part of their normal work, and it's super bright. Mm-hmm. And over the years, I've gotten able to grow it up where it's extremely bright, and then I thought, wouldn't that be nice to do painting with it? Yep. You've probably seen people, take a swab and they'll just write their initials. But students can do fabulous stuff.
Yeah. So you have your students do it, right?
Oh, yeah. We have the Lux Academy Awards every year, Vincent.
Nice. I like that.
Oh, yeah. And I give them prizes for their artwork. But what you're talking about is the work that my wife does with what we call Lux Portraits, where she's able to, look at a drawing of someone or an artwork or a photograph, and she's able, with a fine paintbrush, to use that culture and paint onto the surface of the nutrient media of the plate. And you can't really see what you're doing, can you?
Right.
So if you go tilt the plate back and forth, light reflects off the surface, and you can see sort of where you've been. And if I were to show you the first plates that Jennifer did, you'd say, well, okay, I could probably do that. But you look at what she does now, and no, you couldn't do that. It's really quite wonderful. And it's just practice makes perfect.
Do you ever try it yourself?
I have blunt instruments for hands. I certainly have written microbial haikus, and I tend to like to write words on plates.
Now, your wife is not a microbiologist, right?
She is a mathematician.
Mathematician. So do you bring home these bacteria and plates for her to do, or does she come to the lab?
Oh, no, I bring them home. I know probably now I'm going to get a visit from somebody about that, but no,
I don't think it's a problem.
It's not.
It's BSL 1, right?
And I bleach things before I put them down the drain.
And then you bring them in and put them in the incubator?
Oh, yeah.
And then you bring them back to show her, right?
Right. Now, I want to point out that some other people have done some wonderful work with this. I don't want to give you the idea that I'm the only person who's done this. I mean, lots of people. Simon Park in England has done a lot of this kind of work. But Hunter Cole in Chicago is a fabulous artist that's done stuff that I can't do with some of this. It's well worth your time checking that out.
Okay.
Of course, Susie Wiles in New Zealand. She'll use these quite large square plates that are they're like $12 a piece. And then they'll actually create a large montage and then kind of paste them up on the wall to make large installations.
Right. So that reminds me, a couple of years ago, we brought some of these artists to ASM Microbe, right? Mm-hmm. And they were part of a symposium. Do you remember?
The one I ran?
Yeah.
That one. Yes.
Yes. I was interested because I tried to encourage that to be done by others, and I'm glad to see it happen. And one of the offshoots is that we still have some of the artists here today manning booths, right, and selling their wares, which is really good.
Oh, it's some of the this idea of mixing science and art is not strange. It's beautiful.
It's great.
It takes you back as far as you want to go. I mean, Leeuwenhoek did some of this work. Of course, Fleming did.
Yeah,
and it's fun.
And I remember that the piece we used on TWiM was a portrait of Elio.
Yes.
Right? It was very nice.
Yes.
Lovely.
You've had people paint your portrait in bacteria, though, as I recall.
I did. One year for the Auger Art Contest, someone painted me, and it was lovely.
Yeah.
It was just beautiful. I really thought it should have won. It didn't even come in the top 3, and I still think it was the greatest Auger Art creation of all time.
Okay.
Because I'm biased, of course, because it was me. But this year were some amazing ones for sure. No one has tried again in my visage, although they should, because maybe they'll win someday. I want to ask you 2 more things.
Certainly.
First, when you came, you put a tardigrade on your microphone stand. It's very cute. So what's with the tardigrade?
Well, tardigrades are kind of my spirit creature.
You got a tattoo, right?
I do have a tattoo of a tardigrade.
Nice.
And in Latin it says survive through it, because tardigrades can undergo cryptobiosis, and in undergoing cryptobiosis they can spend decades in this dried state. In addition, they are an extremophile's extremophile. So I love the idea that they can live through anything. Personally, I'm interested in the microbiota associated with tardigrades because if they undergo cryptobiosis, what happens to the microbiota?
Right. So you have them in your lab.
We raise them, yes.
And you study their microbiome.
One of my students is, in fact, the official tardigrade wrangler.
How do you grow tardigrades in the lab? What do you do?
You can't grow them completely aseptically. What is the term where you don't have perfect control? But we feed them single-celled algae. The species that we work with live on single-celled algae.
Okay.
And in pond water. And what's really interesting, Vincent, is that if you put a tardigrade, and they're tiny, you can't really see them very well with the naked eye, they'll crawl all over the algae and it'll tangle up. So you actually know if you have tardigrades really quickly because they don't like to walk on plastic. Instead, they'll crawl on the algae. What
is a tardigrade actually? It's a eukaryote?
It is a eukaryote. It's been some wonderful stuff on it phylogenetically. I can't pronounce the name of but it's been through 5 worldwide extinctions, mass extinctions. So I think they're a good thing for anybody in academic to use as a spirit animal, don't you think?
I love it. I think it's great. Yeah.
Yeah.
It looks very nice on your microphone stand there. But this is a multicellular animal,
It
right?
is. I know. People are really surprised. And I said, there are some eukaryotes I like. This is one. My wife and children are 2 more.
Any others?
My brother.
All right. I'm getting the picture.
Yeah.
One last question. What's next for you? Are you ever going to retire?
I don't want to. My dream is just to have a little lab where I can putter around and do what I want to. But I don't think they pay you for that. So I'm going to have to hit the lottery or something.
Well, you can continue to teach and do research. I'm sure you could do that for a long time.
Oh, I would like to do that. But, I don't – and when I say I have a large class, it does not sound large to other people. But, you get used to what you have. So my intro class, and I'm not happy – that's 48 students, 3 lab sections of 16. And I wish it wasn't so big because I want to be able to deal with these students one-on-one. I really do. And that's what I like about the micro class. And it's what I like about my little Symbiosis course too. To get a chance to really hear what they're thinking and what they believe.
Sounds like you'll stay at Puget Sound, right?
I think they can't get rid of me now, right? So there I am. And I'm at an age now where it would be very hard for me to move anyway. And, Jenny seems happy enough working at the University of Washington at Tacoma. So we've got it's a nice part of the world to live in. Have you been to Tacoma?
I have not.
I'll have to get you out there and show you some of the stuff down at Reconciliation Bay, where the large fibers that'll freak you out.
Cool. 4 millimolar hydrogen sulfide.
I know. Isn't that amazing?
I won't forget that.
But I have it. You've probably seen this. I don't know if how's your Latin?
It's not good.
So on my arm, and it was funny when I got my first tattoo a few years ago, and people quietly contacted my wife asking if I was having a midlife crisis. And she said she liked it much better than a red sports car, which I thought was a good answer. So what this says is Avite Pawile Domini, with a diagram of a bacterium, and that means all hail the small masters.
That's another good title.
It is, all hail the small masters.
Excellent.
And a student saw that one year.
Yeah.
And you will recognize the artwork.
There's a
That's Vaxed. That's Maggie's work.
Ah.
And it says that I'm a microbial badass because that is what that student called me.
That's a microbe there, right?
It is.
Okay. It's very nice.
Artistic license.
Yes, of course. Mark, is there anything we haven't talked about that you would like to talk about?
Yeah, I think that if you're going to do research, if you're going to teach, you have to have a real passion for it. And I had someone I really like and respect once tell me that I needed to be less markish. And I understand what that person meant, and I adore that person, so I wasn't offended at all. But I think what works best with everyone is being genuine. I know it works well for students when you're teaching. There's no substitute for you being genuine. And students will always respond well when you do that. When you say, I don't know, I have colleagues that are afraid to say that. Students don't have a problem with that at all as long as you're excited about finding out the answer. I can't think of a better job than doing science.
Neither can I.
I mean, I get to find out things that are new and interesting every day. And in microbiology, it's new and exciting every day, isn't it?
It is. And I think you are yourself. You're being genuine. You're Mark.
Yeah.
No problem.
Yeah. Yeah, I suppose I could amp back on the coffee here and there. But the point is, I really adore what I do. I think the world of these students, I've learned so much from them. The way that they see things differently. We learn so much together that way. Because truly teaching, and that's at the bench doing research or in the classroom, it's a two-way street. It's not somebody sitting on one end of a log and the patient, there I go, and the student on the other. It's a collaborative process. The problem is we only have so many hours in the day, so you have to love it. And then it's not work.
That's what I tell everyone. It's not work if you love it, right? And it's not a job. It's a career. Yeah, I totally agree.
I would add it's a privilege to me.
It is. It is. I agree with you. All right, that's TWiM. And if you listen to podcasts on your mobile device, that means a phone or a tablet, you usually have a podcast player. You can search for TWiM, This Week in Microbiology, subscribe, and you get every episode as we release them twice a month. They're free, and we would like you to do that. It would really help us. And if you like what we do, consider being a patron. Support us. Go to microbe.tv/contribute, and you can find out different ways. For example, you could give us $1 a month, and that would help us to travel and do other things. And, of course, questions and comments to TWiM at microbe.tv. My guest today from University of Puget Sound in Tacoma, Washington, Mark O. Martin. Thank you so much for joining me.
It was a pleasure to be here.
I would like to thank ASM, the American Society for Microbiology, for their support of TWiM, and Ray Ortega for his technical help, and Ronald Jenkins for his music. RonaldJenkins.com. I'm Vincent Racaniello. You can find me at virology.ws. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.